Hi, good afternoon, everyone. My name is Chih-Hsuan Wei from National Library of Medicine. Today, I would like to talk about recent research about the for gene set analysis using large language model, leveraging the contextual information. Along with the increased studies from the mRNA expression, genomic data, and also the cancer data, more and more high-throughput omic data about transcriptomics, proteomics, and single-cell sequence has been produced. And such data come with the random gene sets. And here is an example. It's a gene set obtained by whole genome long reading sequencing from a study for the genetic subtype of P-progenitor acute lymphoblastic leukemia. And with such gene set, we are able to analyze and categorize them to based on their functions to pathways or ontology. And with those function annotated that we can apply to the following applications, like to recognize the regulation between genes, and also to find the transcription factors, especially the master regulators. And further, we can target the therapy for disease diagnosis. And here I'd like to give some examples recently published. There are some representing studies that are based on the gene set analysis. An example for the disease therapy, the biomarker or the map pathway in gastroesophageal cancer has been recognized. And also, Besmil has been reproduced for human skin inflammation treatment. And also, gene set analysis has been applied for a single-cell analysis to unveil the coordinated global transcription. And I will give you some introduction to the related works, including the traditional enrichment analysis and also the large language model-based approach has been applied to this task. Gene set enrichment analysis is a very popular method, including GSEA, G-profiler, and enrichers. And those methods provides the output data with statistical significance and to tell the enrichment functions and the description p-value to tell the relevance and the gene list of the input gene set. However, those methods are highly relying on existing domain databases, which means it's hard to discover the new data. And also the output results is extremely huge and can be fragmented and with weak transparency problem. So that recently a paper applied a prompt engineering on this problem. The input is a gene set, and they applied a chain of thought approach with the instruction for task, confidence score, and the format with the analysis methods, and also one-shot example. And based on that, the method provides the proposed the enrichment terms and also the process about this, and also the confidence score. On the right is an example. Given a gene list that they propose the functional names and also the interpretative text. However, we discover some of their results are not aligned with existing domain knowledge. That may be due to the hallucination. According to this, we recently proposed a new method named a gene agent, which is also a large language model-based approach to verify the output, mitigate the hallucination. And it maintains model transparency by using clear explanatory analysis. And we also review the results and learn that all the results is aligned with the existing enrichment knowledge, but still some of them are not aligned with the experimental condition because those information wasn't provided and wasn't considered to tell the method about the gene set analysis. That's the reason why we proposed the method contextual gene set analysis to this work. This is the first of its kind framework that integrate the experimental condition or statement to tell how the gene set was generated to verify the domain-specific knowledge to provide the context-aware analysis. In addition to the input gene list, the user can provide the context, which is the experimental condition telling how this gene set was provided into the CGSA, and the result will be a list of pathways or functions and also the confidence score. And here is the flow chart containing 4 steps, and I will describe step by step. The first one is a gene cluster detection. Given a list of genes can be differentially expressed genes, and we use a string to extract the interaction network, and use an animal to which is an age enrichment approach for community detection to group the genes to sub gene sets, because we believe that those genes are close to each other are functionally related. And the second step is the gene enrichment analysis that we apply traditional enrichment analysis to generate the candidates and based on their functions. And the third step is that we started to leverage large language model with the contextual information. The first step is to clean those pathways that are not aligned with the condition, experimental condition. After that, we summarize the results and come up with the relevant score that can be, though the pathway can be ranked, and also provide the discretion to tell why this gene set, why this functions has been selected. And I'll provide an evaluation about our results, and also will give a case study. Actually, there is no existing data sets containing gene sets also with the experimental condition, so that we collect that by ourselves from PubMed by query gene set enrichment analysis, and we look at the data from October 2022 to June 2024, and to narrow down the scope to PMC open access only with supplementary tables. And then we use PubData to narrow down the scope to contain the articles with tables with supplementary tables with more than 50 genes. Then we manually review those articles and collect about 102 differentially expressed genes, and containing 900 genes in average, and 10 diseases, 19 diseases, and 10 mechanisms, including human mouse. And actually, the manual evaluation is very expensive and cannot be done on large scale. Thus, we use AI to evaluate. We use a MassCBD, which is a constructed pre-trained model transformer, to calculate the similarity between the functions recognized by the methods, and also the functions listed in the paper recognized by the authors. And we use different threshold to cut the results. And we find that CGSA achieved a better performance, a harmonic means of accuracy and hits with 87.8%, which represents a 30% increasement than all other methods. And one thing need to be mentioned is only CGSA in this pie chart, in this chart, utilize contextual information. Thus, you can see a very big improvement to the performance based on the context information. And also, we ask to curators, to biologists, to curate the CGSA results only, but not other methods. And they provide a 90% inter-agreement. And according to the human variation, CGSA achieved an 82% on accuracy and 87% to hits, and with a harmonic mean of 85%. And this result is consistent to the AI evaluation. And we did an additional experiment to replace the backend large language model to other methods, to other models, including GPT-4, 3.4, 3.5, and Llama 3.1. And you can see that the performance using 4.0 is higher than all others. And we did an ablation experiment for the gene cluster detection step. And the performance dropped about 12% without using the cluster approach. And I like to give a case study provided by one of our authors, which is a biologist, about the therapeutic resistance of the melanoma. And we analyzed the adopted genes expression in melanoma evolution to identify functions selected for therapeutic resistance. We have two gene expression data. One is from the responders. One is from the cells that responds to the tumors, and the other one is from the cell that doesn't respond to the tumor. And we compare their expression results and find the difference and collect those gene sets that representing the responsive mechanisms. And also we can find the other side for the non-responsive mechanism gene sets. With the two gene sets, we use CGSA to find those pathways or functions and also categorize them based on their categories to 7 different clusters. And the size of the nodes represent the genes, the gene numbers in the functions and involved in the functions. And also the color of the relevance score represents the importance. And we can see that some of the pathways or functions are highly representative to the gene sets. For example, to the gene set R, the function of the transcriptional adoption and the immune response are consistent with the effect immune activation triggered by ICB treatment. And also, the other side, the cell cycle exhibit the dominant enrichment and also the proliferation pathway and suggest ongoing tumor growth despite the treatment. And the conclusion, CGSA bridged the gap between the traditional enrichment tools and also the large language model by incorporating biological context into gene set analysis. And it achieved the state-of-the-art approach, state-of-the-art accuracy, and demonstrate the utilities in real-world use case. In the future, we will explore how prediction are influenced by different aspects of context to improve the accuracy, and will expand the species to non-human mouse. And we'd like to thanks to the contribution to all of our authors, and also thanks to the colleagues in National Library of Medicine for the discussion. And thank you so much. Any questions?
All right, thank you very much. And we do have some time for questions. So, stay where you are, Crystal. Pass this.
Thanks, great talk. So I've tried the method that you mentioned, the previous method, and the main problem I found with that was as soon as your gene list is beyond a couple of tens of genes, it can't kind of contextualize everything, and it comes up with a summary for just a few of the genes. So is your method able to overcome that? And when you have, let's say, 900 genes, you mentioned often,
yeah,
is it able to say something about all of those genes to the same extent that, say, G-profiler would?
Yes, it works. Actually, the example I show in the slides just a few genes, but actually the real cases, the genes more than 100, even 1,000, that is frequent. So even in the case that we are able to do that and categorize them and summarize the gene set and compare with the results found listed in the paper and realize that the results is very consistent.
So is it the clustering you do that allows you to achieve that?
Yeah, actually, we have to first cluster the genes, and then based on the cluster, in each gene cluster, we do we do gene set analysis, and based on those find those functions and pathway found by the methods, we compare with the functions listed in the paper, and those results are very consistent. So which means that if user use CGSA to do that, yeah, that'll be save a lot of time to the gene set enrichment analysis.
Okay. Another question here?
That was a really interesting talk. I'm not sure you might have answered this in part, but I wondered if you looked at doing it without the clustering. Because you can do gene set enrichment analysis just in a normal way. Is the barrier that you won't be able to use the LLM afterwards, or would you be able to take that out to see how important that is to the process? How does it impact your performance if you don't have the clustering?
Yeah, actually we have slides. Maybe I didn't discuss that.
I may have missed it.
Yeah, actually the performance drop about 12% without using gene cluster, but comparing to the performance without using contextual information, that drop about 30%. So actually we can say that half of the improvement is because of the cluster, and the other part is because of the large language model.
Okay, that's great. Thanks.
Okay. Are there any other questions? Okay, so it's now thank our speaker once again.